Hello, everyone. This is Dr. Kellen and welcome to another episode of Inflammatory Content, the podcast about immunology and biomedical research more broadly. I'm here to interview world-class researchers, to broadly communicate the cutting-edge work going on in their laboratories, and to get to know the people behind the science. So sit back, relax, and let's dive into some science. And if you enjoyed the show, please consider sharing the episode, leaving a review, or donating to support our work. Thanks for listening. Before I introduce today's guest, I have some personal career updates to share. I officially started my postdoc in the Gala lab at UC San Diego. Woohoo! So yes, I am doing my postdoc in the same lab that I did my Ph.D. in. Unconventional, I know, but I thought very hard about this decision and ultimately decided that staying was the right choice for me. I also made another unconventional decision to submit an NIH K99 Pathway to Independence Award as a first-year postdoc. I just submitted my grant, and I learned a ton from the complicated application process and plan to record a podcast episode about it sometime soon, so be on the lookout. With that, on to today's guest. Today our guest is Dr. Aaron. Aaron is a postdoctoral researcher in the Mulligan lab at NYU Langone Health in New York City. He and I go way back. Aaron earned his Ph.D. in biomedical sciences at UC San Diego and did his thesis work in the lab of Dr. John Guatelli. Aaron was just finishing up his Ph.D. when I joined the program in 2019. Aaron is a virologist and immunologist. During his Ph.D., he worked on Zika, Dengue, and HIV. As a postdoc in the Mulligan lab, Aaron has been focusing on Mpox, formerly known as monkeypox. Mpox is an emerging infectious disease and represents a significant threat to global health. I am very grateful that Aaron has taken the time to come on the podcast to educate us about Mpox and his latest research study. You can find a link to the Mulligan Lab website wherein Aaron is a postdoc and references to the papers we discuss in the show notes. A quick note about the episode's sound quality. While most episodes of Inflammatory Content are recorded in person, this one was recorded virtually. We had some minor connectivity issues during the recording, so I apologize for the less than ideal sound quality at times and the slight delay between our microphones. So without further ado, please enjoy this conversation with Dr. Aaron. All right, Aaron, welcome to Inflammatory Content.
Well, thanks. Thanks for having me on. Excited to chat.
Yeah, excited to catch up and hear what you've been working on and learn more about you and share it with the community. So I'd like to start with your training history and what got you into science and maybe a little bit about undergrad, grad school.
What got me into science, it's been something that I've always really enjoyed and one of my favorite subjects growing up and everything. I got around to undergrad. I majored in biochemistry and molecular biology at Michigan State. And while I was there, I worked in a lab doing some cancer biology work, specifically focused on microRNAs. I was really fortunate. I worked with a grad student there who had a lot going, and I was able to get on a couple of papers during my time there. And I think that definitely helped set me up when I was looking at grad school. By the tail end of my undergrad years, I was a little bit bored, I guess, of the cancer work. It felt like a lot of the fields had gotten into very specific niches. And I started to look more towards infectious disease work, particularly when I was interning down at St. Jude Children's Research in Memphis. I met with a few different HIV physicians down there and started getting interested in HIV work. And so when I went to grad school, I was looking specifically at HIV folks. And ultimately, that's what brought me then to UC San Diego and working, I would say, with a few different people. Two that I really came down to were either Shane Crotty, who's of course still at La Jolla Institute, and John Guatelli, who's at UCSD. And John was always and still is very much on the virology side, and Shane, of course, is much more on the vaccinology, immunology side. And I found both of their work really fascinating. Shane's lab is a bit bigger than John's and has a different dynamic. It's very high energy, a lot going on at all times. John's was a little bit smaller. He was around a lot more. He's towards the tail end of his career at this point. Shane is very much in the peak.
Especially after he
Yeah.
rotated, right, with COVID.
Yeah. No, I do frequently think about what the arc of my career would have been if I had stuck with Shane. It would have been quite different because, the pandemic came at the tail end of my grad years there. And that was a big thing that altered the trajectory of what I ended up doing. Because when I was working with John, I was focused strictly on HIV. And if the pandemic hadn't happened, that's what I just would have done. I never probably would have switched out of HIV. But with the pandemic, came SARS-CoV-2 and this brand new virus that, coronaviruses are always under research. No one really cared other than a small group of people that were super dedicated for decades. And so there's a lot of these really big open questions that were left. And it was kind of like, what about this simple question no one's ever looked? It's like, oh, great. That's an interesting question, low-hanging fruit. So I started digging into more respiratory viruses towards the tail end of my grad years. And then when I was looking at switching into postdoc, I was focused on trying to blend vaccinology as well as immunology with the virology of it all. And I was particularly focused on SARS-CoV-2. And ultimately, that's what brought me to NYU, where I work with Mark Mulligan now in the NYU Langone Vaccine Center. And there's a few groups that are in that umbrella, but they're focused much more on the vaccine immune side of things. So it's been a really big change for me doing much more immunology. There are still frequently times where I'll ask, I've got a co-mentor now, Raman Haradi, who I'll ask him questions and it's like, okay, what about this? And then it becomes this whole 20-minute conversation. I'm just like I thought it was going to be a simple answer. Like, dang, okay. So still very much getting used to the immuno side. I think long-term what I'm looking towards is staying with academia and having my own lab and being on that interface between viral immunology and using that to use virus basic knowledge and develop vaccines that are reliant on either attenuated viruses or viral vector systems and the like. So that's been really coming out of the work that I've been doing now, particularly on Mpox, or this is formerly called monkeypox.
In your PhD, that was much more basic virology, right? You were doing computational systems biology work. Can you just say a little bit about the major takeaway of your thesis from your PhD?
Yeah. So my PhD kind of went on a pretty winding road. Basically, what we were trying to accomplish was there was a junior faculty in the group, and she's still there, Mary Lewinsky, who developed this inducible HIV system within a Jurkat T cell line. It had some advantages in that anytime that you're infecting a cell line or any host with a virus, you're generally not getting a pure population of infected cells. The MOI really has a major impact on that. And so you do start to have weird things that can pop up too. If you're saying, okay, I'm going to put so much virus that I guarantee every cell will be infected, you're going to get cells that are multiply infected and start having slightly weird biology compared to, just a normally infected cell. In developing this inducible system, what was really nice is that Mary had made this cell line that basically we could add doxycycline, turn on this dox-inducible HIV expression cassette, and all these cells then would effectively be infected. But they hadn't gone through the initial steps of viral entry and the priming and then integration into the genome. But basically, it was as if starting from post-integration, you then have turning on of the viral genome. So that system was really handy because then what we could use that for was doing proteomics. Because anytime you're doing bulk proteomics, the problem that you run into is that it's an average of everything. And so if you have half of your cells infected, the other half not, then if you're trying to look for perturbations in the proteome, it just doesn't really work. You need as many of the cells infected as possible. And what we could see is that with our inducible system, we were getting usually 90-95% of the cells would be expressing the viral genome. So the whole point of this system was to try then to take this and see if we could look at the spatial proteome and try to understand how exactly one of the accessory proteins of HIV, NEF, is able to alter where proteins are in the cell. Because where a protein is in the cell has an impact on how it functions. It impacts what's around it, the other proteins, and how well it can actually perform its job. So we use this system then to try and interrogate the spatial proteome of this T cell model. And towards the very end of my PhD, I had a committee meeting, and on my committee, I pulled a good group of people that had expertise across different areas that were relevant to the project. And for the proteomics side, I had David Gonzalez, who's still at UCSD in the pharmacology department there. And I was going through all this data, and one of the findings that we had when we had gone through, done these spatial proteomics projects and everything, and that had been partnered with Nevin Krogan up at UCSF. We got through all this, we did all the analysis, and I was digging into, it looked like there was perturbations of the peroxisome, and that NEF was doing something to the peroxisomes. And there's a bunch of literature that describes potentially antiviral functions that can be tied into the peroxisome. So we thought, okay, this is new, this is novel, no one's really looked at this could be interesting to dig into. And, I was going through the technical details of the proteomic analysis, and this is at a committee meeting, and David says, well, hold on, you use which software to do this? And I said, oh, this particular software to analyze the proteomic, taking the hits from the mass spec machine and then mapping that onto actual proteins to do the quants. And he says, well, hold on, we've tested that software. It doesn't work. And so I ended up going back in, having to reanalyze all of the raw mass spectrometry data. And at this point, realizing this all has to be redone. So I go back in, I redo the entire computational analysis from scratch, and I'm getting different results. And also at this point, we had submitted the first draft of the publication to a journal. We got some pretty rough reviews back. And at this point, I was ready to transition and to be heading out the door. I was starting to interview for postdocs. Things were in progress. So with all of this coming together, I scrambled a bit. And what I was able to ultimately do then was take this project. And what's nice about HIV is that it has been studied extremely thoroughly for decades now. And it was one of the things that I was getting a little bit frustrated with in the latter years of my PhD was a lot of the work that you do within HIV, you're not getting these splashy big findings anymore. Generally, you are digging down into, sometimes specific residues where you're modeling out and you're saying, okay, residue 424 on this protein is interacting with residue 217 over here. And how do we model? And you're doing very nitty gritty work. And that is taxing and tedious at times. And so I was getting a little bit burnt out on that, but it did serve a nice purpose at the end because there was so much already known about HIV that this spatial proteomics actually works in that, okay, well, there's a lot of things that this should be turning out that we've already shown in the field. And so what I was able to do then was transition the project more towards a computational methods. And that really was ultimately focused on then taking work that had been done in this small field of spatial proteomics and saying, okay, 3 or 4 different groups have developed these analytical pipelines. And every single time somebody published a new paper, they had their own new analytical pipeline. And no one had ever said, okay, what if we take these multiple analytics and use them all on the same data set? How does it look? Do you get the same thing out every time? And what we found was that when we use these different machine learning algorithms to analyze our data, it gave us some variable results in how well each of these classifiers were able to spit back out known knowledge for HIV, as well as baseline known locations of proteins within the cell. So while we weren't getting a lot of novel HIV biology out, it was interesting to be able to take kind of this field that was still kind of relatively nascent and say, okay, how do these different analytical methods actually stack up against each other? And so I ended up getting out of my PhD a ton of virology experience, but also got a lot of computational experience that was honestly really useful. And even when I'm not doing kind of heavy machine learning work, it's still nice having the computational skills when I'm doing basic analyses, or sometimes in the work that I do now, we get a little bit higher dimensional work and we're trying to push into more single cell as well. So it's all useful still.
Absolutely. Yeah, I think it's a good lesson too for people doing computational stuff to always try multiple models for your data because there are so many model dependent things that can come up.
Yeah. And it's definitely one of the things that I've come to appreciate as I've gotten a little bit more into the sequencing side of things is that there are groups that are dedicated to doing benchmarking and that put a lot of time and effort into helping inform the field on what are the best analytical tools for a given application. And that's really within the sequencing side of things. Proteomics is always lagged behind. It is more expensive. You get less out than with sequencing. And that was really apparent in spatial proteomics that it was a handful of groups that were really interested in it, particularly in a virology capacity. And so, yeah, it's definitely worth looking at the tools that are being used, especially if you're in a field where it's every man for himself. There's not really a clear winner or loser in how you're doing these analyses.
Right. Very important benchmarking work that you did there then. There were a lot of transitions that you had from your PhD to postdocs. Virology to more immunology, vaccinology, computational to experimental, basic to translational, and West Coast to East Coast, right? You were at UCSD to New York. That's a lot of changes. Were you just ready for something different and wanted to switch it up?
Yeah, I've always been a big proponent on the moving side of things on a number of levels of kind of making radical shifts at each point of the career.
Whereas me, I stay in the same place forever.
And yeah, there's benefits to each side of it. I grew up, I did my undergrad in the Midwest. I went to the West Coast for grad school. I'm out here on the East Coast for postdoc. I do probably want to stick around New York ultimately for academia, but I think there's a lot of value in bouncing around and getting different takes on problems that have different sides to them. I was at a conference over the summer. I was at American Society for Virology, ASV, and there was a pox virology session that I sat in on, and it was really interesting. Everyone in the room was very, very up on all of the pox virology. They knew all of the pox virology background. They were very well-versed in all the virus side of things. And then when more immuno stuff would come up, you could just tell that the expertise in the room was further back from where their virology expertise was. And it was the same thing when I presented at ASV this year and presenting on this Mpox vaccination work. I was in a vaccine-specific session. And in that session, I got a lot of questions on the immuno side of things, but there was not a lot of questions on the actual virology side. And so it was interesting to see that there wasn't really a clear crosstalk. There wasn't a lot of people that were fluent on both sides. And I saw that too in HIV. My advisor, John Guatelli, he's a very accomplished virologist. He's very good at virology. And I remember he would say, though, that he would openly admit to his colleagues, he'd say, my immunology is very poor. So he'd ask basic questions and stuff. And I think that that's the case of a lot of people. There isn't that crosstalk of the virology and the immunology. And so I didn't strictly set out to cross-train, but I've now come to really appreciate the value that I have gotten in this cross-training. And, I think I'll always be a far more proficient virologist than immunologist, but, there's still a lot that you can get out just from having an understanding a little bit beyond surface level. And it was the same thing with computational versus bench science. I was doing a ton of bench science in my grad years, but I got to have a lot more hands-on with the computational side than I might have otherwise had if I was sticking solely to the bench side. So the computational one is one that I've always talked with people and said that I think the more that you understand both the computational, both the dry lab and the wet lab side, the better off you are. Because even if you're not doing the other side of it, at least you can converse with your colleagues, your collaborators in a cogent way. You're not saying to them, okay, here's the data, just hand it back to me, and they hand it back to you, and you have no idea what they've done. At the very least, you can then design your projects in a way that will make sense for how the analysis is going to be done. And likewise, if you're on the computational side, you can say, hey, this is a consideration that you need to make when you're designing the project so that this computational method makes sense for what you're doing. So I've come to really appreciate the value of cross-training. And it's something that, while it's not something I've always aimed for, I keep falling into it and then appreciating that I've done it.
Yeah, I couldn't agree more with that, with the computational stuff. Let's talk a little bit about what you're doing in your postdoc now. So most people have probably heard of monkeypox, or now more commonly referred to as Mpox. Can you briefly talk about Mpox? What is it? Why is it a problem? Why should we study it?
Yeah, so to back up a little bit from Mpox, just briefly, I think most people, when they think of pox viruses, what they're most familiar with is smallpox. And after the eradication of smallpox in 1980, that the official declaration by the WHO, the pox virus field was, I think, a little bit out in the wind. And really what motivated continuing interest in the pox virus field was this idea of essentially bioterrorism, the idea that somebody could manufacture smallpox again, or that they could create an alternate version of smallpox that could then spread throughout the population. And so there was this continued interest in studying pox viruses, even though there weren't major global pox viruses. But at the same time, from the 1970s on, there was a clear presence of Mpox, or as it was formerly called monkeypox, within humans, within a central swath of Africa. So really you had the kind of Congo Basin and kind of Central Africa, and then as well as the western coast, kind of the northwest coast of Africa, where you saw Mpox. And there wasn't a ton of cases. There just wasn't a lot of human spread within those countries, even where it was endemic. But in the last 20 or so years, we have seen that there's been an uptick of cases. And the thought is that most likely it's coming from, as we have kind of waned off of having smallpox vaccination, the larger proportion of the population is not smallpox vaccinated. And the nice thing about the poxvirus family is that they're all pretty similar on an antigenic level, which means that if I vaccinate you with one family member of poxviruses, you're probably protected against most of the family members. So that was the strategy that was used for smallpox, and we used vaccinia virus for that. And as we continued this research into smallpox virus vaccines, it was generally relying on these vaccinia vaccines. And they were all generally attenuated, but like early smallpox vaccines, what we consider first generation, things like Dryvax, the side effects were rough, to say the least. It was something like 1 in a million, 1 in 2 million people that were vaccinated could possibly die. Now, that was an acceptable trade-off to us because smallpox was roughly a third of people that got smallpox would die. Right. It's always the case in vaccinology and in therapeutics in general of how severe is the pathogen that you're trying to fight, and that will really inform the kinds of side effects that you're willing to tolerate. So after the eradication of smallpox, these subsequent vaccine generations, the second and now we're on third gen, we have a far lower tolerance for side effects that are severe. And so second gen vaccines is really what we have relied upon up until this 2022 outbreak of Mpox that spread pretty much globally. And within Mpox, there are two distinct clades. There's clade 1 and there's clade 2. Clade 2 is what drove the 2022 outbreak. And generally, clade 2 has a lower pathogenicity to it. But that outbreak really caught people off guard because even if we had seen increasing cases within these African nations where Mpox was endemic, we weren't really seeing spread beyond those countries. And so when that 2022 outbreak rolled around, it caught a lot of people off guard. And it also...
And this outbreak was, you saw the majority of cases in the U.S., right?
It ended up being, a little over 90,000 cases. And I think it was something like 40,000 of those or so were within the U.S. So we had quite a few cases. We are a large nation, so it's not shocking that we account for a decent amount, but it was surprising how quickly and easily it was spreading. We can circle back later to what ultimately curbs that outbreak, because there's a few different theories on how that ended up fizzling out. But when the outbreak rolled around, we had a bunch of these second generation smallpox vaccines, primarily ACAM 2000, that were sitting in a strategic national reserve. So again, this goes back to this idea of bioterrorism, the thought that if somebody were to weaponize smallpox, we want to be ready. And so the US has an entire strategic national reserve of this ACAM 2000 vaccine. And it is a more attenuated version of the first generation vaccines, but still using vaccinia virus. But because it is more attenuated, doesn't necessarily mean that it had no side effects. And so in the last 10 or so years, there's also been interest in this more recent third gen vaccine called MVA-BN, Modified Vaccinia Ankara, and the BN is Bavarian Nordic, who manufactures it. The trade name that we have here in the US is Jynneos. It also goes by Imvanex or Imvamune in other countries. And the major difference between vaccinia and MVA is that vaccinia will still replicate within humans and within mammals broadly. But MVA will not replicate. Basically, it triggers what we call an abortive infection. So this virus will go, it'll infect cells, it'll get inside, it'll start the replication process, but at some point within that process, it just stops. It's not able to go on and make new viral particles from that infected cell. This makes it generally a safer vaccine, particularly in people that are immunocompromised, because it's always a trade-off that we have with these viral vector vaccines, even attenuated ones. You run a risk if you put it in an immunocompromised individual that their immune system won't be able to handle it. So when the 2022 outbreak rolled around, we began to rely on this MVA-BN, Jynneos, and there was definitely a major shortage, though, and that created a couple of interesting branches in how the vaccine was used. So the vaccine, as it's FDA approved, is a 2-dose vaccine. Each dose is subcutaneous, and it's 28 days apart. When we had this shortage, the goal was to get as many vaccines out as possible. And so they said, okay, we are going to focus on first doses. We will circle back to getting second doses when we have more supply. And after about a month and a half of vaccinating folks, there was a decision made based on some previous work in the literature. It's a paper or two. It's not a lot, but it was some smaller trials that suggested you could use a 1/5 dose intradermally injected as opposed to the regular subcutaneous dose. And so the question that we had when the outbreak rolled around and the vaccine campaigns were going was, okay, can we see if these smaller doses delivered intradermally are giving the same immune response in individuals with HIV? And this was particularly important because Mpox in that 2022 outbreak was really focused primarily within the LGBTQ community, particularly within men who have sex with men. It was kind of the same thing that we saw with HIV, where you had a certain amount of sexual transmission occurring, and direct physical contact is really the most efficient way for Mpox to spread. Because of that, once you get into a particular network, kind of a direct contact network, it sticks within that network. And so it just so happens that you had Mpox get into this MSM network, and then it was able to work its way through that network. And we saw cases that would stretch out from there, but it was primarily within this network. And you do have a higher proportion of HIV within certain portions of the MSM community. And so this was a directly relevant question to the at-risk people that were being vaccinated. And particularly, we saw that a lot of the Mpox cases that were severe were within individuals that have HIV. And the vast majority of the deaths that we saw from Mpox in that outbreak were from people that were pretty significantly immunocompromised due to HIV.
So you wanted to assess on a large scale the immune response in individuals that did and did not have that original vaccinia vaccine, right?
Yeah, and so actually that was something that kind of came along a little bit later. Prior to the outbreak, I was not a pox virologist. Pox viruses are DNA viruses. I've always worked with RNA viruses. The two worlds are quite distinct from each other. And so I had very little knowledge on pox viruses, and most of the group had, sparing knowledge on it. So there were a lot of considerations that we didn't really think of until later on, once the trial that we had established was going. And yeah, the thing that was most interesting that really came out of this was that when we ultimately looked at our data, we segregated out two groups. So folks that had received prior smallpox vaccination and those that were naive to generally pox viruses as a whole. As you can guess, people that were vaccinated against smallpox, that was generally folks that were born pre-1971 in the US, 1972, right around there. That varies a little bit, but generally 1980 and before birth. And then after 1980, generally people that were born after that weren't getting smallpox vaccines. There's some occupational recommendations, but generally you're not getting it. So we had this older cohort that was getting the exact same MVA-BN Jynneos vaccine as this younger cohort. And generally the thought within immunology is that an older group of individuals that are receiving the same vaccine as a younger group, the older folks are usually having a poor immune response. Right. You start to get in immunosenescence. And what we were finding really interesting is that all these folks that had received prior smallpox vaccination, we saw that basically after Jynneos, that their immunity was pretty flat. There's a little bit of tailing off as time goes on, but up to 1 year after getting Jynneos, both doses, their immunity was still nice, detectable. They were still seropositive for neutralizing antibodies against Mpox itself. Whereas folks that had no prior smallpox vaccination, within half a year, almost all of them were seronegative for neutralization. And actually one thing that we've recently dug into, and that work, because we submitted a preprint, and that's what you're referring to, that work we've updated now and we're working on submitting soon. We've also added in some B cell memory assays, and we do see that there is, at the very least, B cell memory up to a year out, at least in a proportion of individuals. So there's kind of this question of what exactly is going to be protective, but the lack of neutralizing antibodies after about 6 months really caught us off guard, because when people talk about smallpox vaccination within immunology, that and like the yellow fever vaccine are considered some gold standard vaccines, because decades after you've been vaccinated, you still have detectable antibodies, not just cells. Memory cells are one thing, but having actual detectable antibody titers against a pathogen decades after you've been vaccinated is impressive. So the fact that we went from people that had a decade on, sometimes 60, 70 years after vaccination, clear detectable neutralizing antibodies against Mpox, and then folks that got just Jynneos are basically negative after 6 months, told us, okay, there's a very clear divergence between the quality of immune responses that we got out of the original smallpox vaccines and this newer third generation Jynneos.
Does the field understand the importance of the B cell antibody response in Mpox? I know with COVID, antibodies are helpful to prevent infection, but T cells are really the important factor when it comes to preventing disease. Is it the same story in Mpox?
Yeah, the tricky thing with Mpox is, and in all the pox viruses, is that they have added layers of complexity to them. Again, pox viruses, they're DNA viruses, and so their genomes are roughly 10 times the size of coronaviruses, and even bigger than some other viruses that are typically smaller, coronaviruses being larger on the RNA side. So in SARS-CoV-2, for example, it's kind of nice. Generally, the neutralizing antibodies are really focused on spike, this one entry and fusion protein. The difficulty with pox viruses is that you have roughly 2 to 3 dozen proteins that are involved in viral particle attachment and fusion. And there seems to be probably some level of redundancies across them, but they each probably have some unique features. And so you can have neutralizing antibodies against any number of these proteins, but that may not necessarily give you total neutralization of the virus. And the additional wrinkle that's added here is that the incubation time for Mpox, particularly clade 2, is decently long. It can be up to 3 weeks, so from infection to symptom onset. So generally, when you look at your memory cell response, it takes a few days for it to come up. And so it's not really clear if you have absolutely no neutralizing titers, as these folks that probably got just the 2 doses of Jynneos are looking at. If you don't have neutralizing titers, how well protected are you? I mean, chances are you're probably protected against some level of disease, but it's probably not a total protection if you had these really high elevated neutralizing antibodies that could fight off the infection itself. But it's not entirely clear. There's definitely a role that is played by neutralizing antibodies as well as the cellular response, particularly on the T cell side. But how all of those play into the course of disease is not fully understood, and it's even more unclear when we look at Jynneos, where you definitely have a loss of these neutralizing antibodies compared to smallpox vaccines, where you probably have at least some amount of neutralizing antibodies still around.
There was a big commotion about Mpox 2022 with the outbreak, and people were still thinking a lot about COVID at the time, and there was a lot of bandwidth required for people to consider both of those viruses as pandemics at the same time. And it went away a little bit in the media, but now I see it coming back. I see more Mpox in the news. What's going on now, and what are the strategies for vaccination that people are thinking about going forward?
Yeah, so we're kind of in a bit of a twin problem right now. So that 2022 outbreak was driven by clade 2, specifically clade 2B virus. And as I mentioned, that's the less pathogenic of the 2 clades. What we're seeing right now is this prolonged outbreak within the Democratic Republic of Congo, within the DRC, of clade 1 virus, specifically clade 1B. And the problem there is that whereas with clade 2, particularly clade 2B, we see a case fatality rate of probably anywhere between 0.1, maybe at its upper limit 1%, but probably sub 1%. With clade 1, the issue that you run into there is that outbreaks of that within Central Africa have had case fatality rates of anywhere between 5 and 10%. Now, you're also dealing with difficulties that you have a higher proportion of HIV prevalence within some of these countries and some of these regions. And that kind of complicates getting a rough idea of what that case fatality rate may be in Western nations where you have better access to kind of advanced healthcare, particularly well-equipped hospitals. And so what we do know, though, is there was a recent study that was looking at the role of supportive care, and actually was specifically trying to understand the ability of this antiviral we have that we think probably helps a bit with Mpox, but this study suggested that it doesn't really seem to do much of anything for clade 1. But what that study did tell us is that just the simple addition of supportive care that you might not otherwise have, being able to go into an ER or into an intensive care unit, that can bring the case fatality rate to probably 2 to 3%. So that's not nothing. 2 to 3% is still fairly high. And right now, clade 1B has mostly stayed within the DRC, as well as you're seeing a little bit of neighboring nations starting to see cases. Where it really pushed into the news at large was when we saw a case from travel that went into Sweden. And there's also been a case in Thailand as well. But that really put it on the map again, because the clade 2B outbreak has mostly tapered off, although we are seeing a slight rise in cases, in the US as well as other countries. So that hasn't gone away. We never got to 0 cases on that like we really were in the pre-outbreak days. So that never stopped. And in my mind, there's zero reason to think that this clade 1 outbreak couldn't spread globally like we saw with clade 2. We're in a great spot right now that if we put resources towards controlling this clade 1 outbreak, we could avoid having a global outbreak of it. It is within our power to do that. All you need to do is get more vaccines into the DRC as well as neighboring countries. There isn't a clear set of data showing that Jynneos has high efficacy within clade 1, but to be fair, there's also not really a clear data set showing that within clade 2. Based on all of the immunology that we've generated against pox viruses over the years, it's quite likely that these vaccines are protective. The durability of the protection is definitely up for grabs, but they are probably protective at least within a 6-month to 1 year window. And so clade 1 versus clade 2 Mpox, they are fairly similar on an antigenic level. I think it's something like 5% sequence difference between the two. And a 200 kb genome is a decent amount, but 95% similarity is fairly high, particularly on an antigenic level where some of those proteins have a little bit higher conservation. In that case, I think that there's every reason to think that if you flood the area with Jynneos, particularly if you're using ring vaccination strategies where you're really saying, okay, we have a case, let's vaccinate all their close contacts, that can help reduce the amount of vaccine that you need. And it also gives you then a more targeted approach to the vaccination rather than these more broad campaigns. But again, it's well within our power right now to contain this clade 1 outbreak. Otherwise, the clade 2 outbreak, all the genomic evidence really points towards most likely that it came from an outbreak of clade 2 that started in Nigeria back in 2017. And that outbreak basically persisted until 2019, and then it fell off the radar. Most likely, there just wasn't the proper surveillance going on during the pandemic, and then suddenly we saw this global outbreak. So there was evolution going on during that time that probably assisted the virus in having a little bit easier time transmitting than it had previously. But we don't have to have that happen again. We can prevent that.
Right. Sounds like there's a lot of important global health work that needs to be done, and also just basic and translational research. I know you are interested in starting up your own lab. Is this what you want to work on?
Yeah, what I'm really curious about, and we don't talk about it too much in the preprint because it's a little bit more speculative right now, but what I'm really interested in is this dichotomy within antibody B cell responses, where what we typically think of is that you have the introduction of some antigen. That antigen then fuels a germinal center response. And the germinal center is really where you have the meat of your B cell response occurring. And what I mean by that is basically you'll have B cells and T cells meeting each other. And what's happening is that the T cells are helping to drive this directed affinity maturation, where essentially you're pushing your B cells to evolve their B cell receptors, the actual antibody, into being a higher affinity, a better fit for a given antigen. And, this gives us a lot of power in that, the immune system will generate both low affinity as well as high affinity antibody responses, and they both really have kind of a role to play. But, there's also responses that exist outside of the germinal center and that probably don't get as much of this kind of directed affinity maturation and that are probably just not as high a quality response, but they have their own role to play. And we consider these to be extrafollicular responses. And our data suggests to us that Jynneos is creating an extrafollicular response, and that's why it's not as durable. And the data that we've shown in our preprint is that it's also a less avid response. The avidity, kind of the bulk affinity of these antibody responses against Mpox proteins is far lower in people that only receive Jynneos compared to the people that are getting Jynneos as kind of a boost after having been originally smallpox vaccinated. So that's what we think is going on. And so that's something that I really want to delve into. And and kind of focus more on taking kind of the basic virology experience that I have and kind of melding it with this immunology and seeing, looking at ways of kind of pulling back into the lab and kind of pulling back from the translational and clinical side and looking at, in vitro organoid model systems as well as other, animal model basics, and trying to understand, okay, what exactly is going on with Jynneos and how might we be able to augment that? Is that something that we can then use to pass on to other vaccines and also kind of helping us to understand more specifically on the immunology side of things, this germinal center versus extrafollicular response and how exactly that breakdown kind of occurs and what role each has in giving us a protective, useful immune response.
You said mouse models. Can you study this in mice, or what animal models are available?
Yeah, so on kind of the basic immunology side of things, there's a lot of work that people have done in probing this extrafollicular versus germinal center response. When it comes to the poxvirus side of things, there has also been a good amount of work in animal models. Bernie Moss's group at the NIH is definitely a really big player in this, along with Inger Damon. His group, I believe, is down at CDC, if I recall correctly. Both of them have done a lot of work in trying to understand ways of looking at using animal models to study poxviruses. And Bernie's group has also been really key in coming up with these cast EIJ models. These are cutaneous mouse models that, for whatever reason, they are just very highly susceptible to poxvirus infection. And that's really nice for Mpox because other mouse models are just not very susceptible to Mpox. And so this then gives you a really nice system where you can do these Mpox studies in a mouse model that is highly susceptible, and you don't have to give them like 10 to the 6 PFUs. You can give them more reasonable doses of virus, and you can see kind of a natural course of infection. And you can also, Bernie's group has shown that you can also use these mouse models for looking at vaccine studies as well, specifically for Mpox. So there's been some nice work kind of establishing these models within Mpox, and it's something that I would be interested in picking up and looking at more, particularly on understanding what these models can do for us in understanding the vaccine responses and how that kind of factors into proper protection against Mpox.
Before we move on from Mpox to some more science peripheral questions, let me see if I can summarize your recent findings. So the Mpox vaccine gives you neutralizing antibodies, but they are short-lived.
Yeah.
Unless you are one of the people who was born before the 1980s and had a smallpox vaccine, then you generate the same level of neutralizing antibodies at first, but then they last longer.
Yeah.
And we think that is because they have higher avidity. They bind more tightly. Is that correct?
Yeah. Yeah. So, we think the avidity is part of it. We also think that there might be some component of the cellular immunity that is distinct between the two groups, that original activation and maturation of the B cells probably plays a part in downstream things such as durability and responsiveness to subsequent exposures.
Right. It's not necessarily that the antibody itself, the single molecule, has a longer half-life. It may.
Yeah. There's probably a difference within the long-lived plasma cell populations between these two groups. And that is just something that is harder to study because you don't see as much of those populations within general circulation. Usually you have to get into the bone marrow. And that is a trickier feat to do on a large scale.
Awesome. Yeah. Fascinating stuff. And sounds like there's plenty of things to work on in your future lab. So transitioning to independence, when's this happening? When are you going to hit the job market?
I mean, yeah, it's definitely, it's an intimidating process. A lot that is asked of in a faculty member that you just really don't ever get a lot of training for. in running a lab, you're basically running a small business, and that's just training that you're not really getting. So that's a little bit intimidating. If you're in a department that's actively hiring, you get the opportunity, and it's a really nice opportunity to be able to sit in on job talks where other postdocs that are towards later years or even junior faculty at other universities will come and talk about their work and see if there would be a good fit for the department. And those are kind of a blessing and a curse. Being able to see how other people do it is really useful and informative, and kind of, okay, this is how I should be communicating. This worked really well in this talk, and this didn't work very well in this other talk. So that's useful. Where it's a little bit intimidating is when you see these people that have like a really beautiful through line where they happen to work on the exact same thing as like an undergrad, as a grad student, as a postdoc. And they have like this really beautiful, arc to their work. They also have been like hyper productive, and they rattle off all these accomplishments. And you're just kind of sitting there in the audience, just like, hey, haven't done half of that stuff. Like, oh my gosh, like, am I going to be competitive compared to this person? So it's definitely a great way to get really into your own head. But it's been an exercise in trying to be able to look at the work that I've done and say, okay, where's the compelling story to what I've done and what still needs to be worked on? So the work that is detailed in that preprint is really a lot of serology-based work, and the questions that we still have outstanding are really cellular-based. So it's something that I'm working on right now, and I'm hoping to kind of have put together in the next year or so is really trying to look at the antigen-specific B cells for Mpox and understand, okay, can we identify signatures of germinal center activity versus extrafollicular maturation? And the goal there is basically kind of use this remaining year and a half of my postdoc and say, this is what we think is happening within humans. And now my goal is to pull back from the humans and be able to do much more deeper mechanistic dives using things like animal models and cell-based models like organoid systems. So I'm hoping in probably the next year or so to start getting out onto the market and be getting my name out there and really looking around, particularly New York City, for openings in, particularly microbiology departments. And as part of that, I'm also aiming in February to be submitting for a K grant, these career transition awards, just because it is so hyper competitive out there that if I can bring in funding, then it just ekes my name up the list even a little bit. So every little bit helps.
Would that be a K99 or a...
Yeah, I'm looking probably at a 22. The 99s, I'm coming up on 3 full years in the lab right now. And for the work that we're looking to propose more on the cellular side, the tricky thing with the 99s, and really this is with any NIH grant, is that by the time you have enough preliminary data to start fueling these applications, you submit the application, unless you have a really like knockout project, you're probably going to resubmit. And so from initial submission and then resubmission, and then by the time you actually get the money, it's probably been another 1.5 to 2 years. And so you're still working generally on that project in that time. And so you've progressed quite a bit on that project. And so by the time they say, great, here's the money to do the project, with the 99, the K99 has that 1 or 2 years of postdoctoral dependent time where you're under a mentor still before you transition to the R00 phase where you're independent. And so realistically, it's kind of been drawing out the timeline for me. By the time that I would get awarded that postdoctoral time, if I were to get the award, I would be at the tail end and basically ready to be moving on anyways. So the 22 just made a lot more sense for me, especially the K22s are much less competitive. They're still competitive. You're still, probably a fifth to a quarter of applications will get, funded, but that is, I'd argue, better odds than the 99s, which are very difficult to get. So, and there's also a little bit more grace in the eligibility timeline for the 99 versus 22. The 99, your resubmission has to be within 4 years after you've been awarded your degree, your PhD, and that where that falls, you can kind of argue a little bit back and forth with them to get maybe a few extra months, depending on how you did the kind of tail end of your PhD years. And the K-22 gives you 5 years of eligibility. So I'm comfortably within the range on that. The 99 would be a little bit more of a push.
We caught up last week to set up this conversation and asked you if there are any topics you'd want to discuss when we hopped on this call. And one thing you mentioned was when to kill a project. What thoughts do you have on when to kill a project?
I mean, it was something that I had to deal with my dissertation, that I had that point where the project was seemingly imploding. And it was kind of a question, okay, what do I do here? And I had the option of, I could have stayed for another year or two and redone all the analyses, figured out new biological hits, chased after those, revalidated them, submitted, and, gone through that whole process. But, I was kind of at a point where I was feeling burned out and I was ready to make the next moves. And so obviously at that point, I had invested so much into this HIV project, like there was no getting away from that. That had to be wrapped up in some way. And so the way of wrapping up that project or in the kind of killing it was basically saying, okay, what if we make this a computational methods paper? And that was never the original goal, but it was like, okay, the reviews that we got back on the first submission were basically, why did you make a new computational method? Why didn't you use these other ones and see how those do? And I was like, okay, let's give the reviewers what they want. I will go through and I will test all these other computational methods that have been published, and I will show how all of them stack up in this pretty well-characterized model of HIV. So in that case, it was kind of an example. It wasn't just dropping the project, but it was saying, okay, how do I pivot on this project and still meet my own personal timeline while doing justice to all the work that had been done? And in my postdoc years, I've had some difficulties in projects here as well, that when I came into the lab, there weren't really ongoing projects. There was a lot of kind of small little things that had been done and little projects that would be basically wrapped up with the course of just one paper. So there weren't really a lot of lingering threads to pull on. And so basically I came into the lab with this larger idea of what if I look at coming up with a VLP-based, virus-like particle-based vaccine for SARS-CoV-2 and leverage the vaccine knowledge that's in the lab and use that to play around with this system. And I spent a bunch of time trying to get that system up and working using kind of the virology stuff that I've been doing in my later PhD years for SARS-CoV-2. And the system just wasn't working. It just wasn't working at all. And simultaneous with that, I was also playing around with some work on this viral competition project that was looking at trying to understand the competition between SARS-CoV-2 and influenza and how those two might compete within a host and what impact that might have on evolution. And that project also had some technical difficulties that ultimately stemmed back from some bogus information that I received from somebody that I then found out later was incorrect. And then when I fixed it, the project started working. But at that point, this Mpox project had come along, and basically that picked up steam and really took off. And so that really took off about a year into my postdoc. And at that point, it was kind of like, well, I need to be at a sprint. I need things that are working. I need to pursue this project and put my all into that. So the VLP project that I had, as well as the viral competition, the VLP project, I basically had to say, okay, the system isn't working. And I had about half a dozen ways I could troubleshoot it. But the problem I had was that they were all tedious. And at the end of the day, I could do all these troubleshooting techniques, and they may not give me information that was actionable. It could be that these virus-like particles that they had maybe, a weird morphology. Maybe that was the problem. And that's why they weren't infectious like they should have been. But then the question would be, okay, well, how do I fix the morphology? I don't have a simple answer to that. So I kind of hit a point where I was like, okay, look, I've been chugging away this project, and it's like, I'm not getting a solution. It's not functioning. I was like, I just need to pivot. And so in that case, it was basically, I'm going to kill this project. I'm just going to drop it. In the case of the biocompetition one, I finally got it functioning. And it was like, okay, well, now this project is far behind where the Mpox one is. And maybe I just need to run with the Mpox one. So that's one that I've kind of put in the back of my mind as like, okay, it's something I'm still interested in, but maybe I circle back to it later with my own lab as an idea of, okay, this is something that I can have that's a little bit diverse from the Mpox work. And I now have a solid idea of exactly what I need to do. You know, all the learning and hurdles that I went through are still valuable. It's just kind of delayed gratification. So it's been kind of a variety of different approaches to how exactly do I take these hurdles and move forward. In the case of the HIV stuff, it's nice that I was able to still get something out of it. In the case of the other ones, it's frustrating, of course, having to just totally drop stuff. But at the same time, like in my postdoc years, it feels far more urgent moving forward than it ever did in my PhD years. You know, in your PhD, unless you're like a super phenom, particularly if you're in computational work where you can move a lot quicker, you're going to be in your PhD for 5, 6, 7 years. That's just what it is. That's not going to change. It's nice to have a productive PhD years, but also it didn't feel like okay, I have to get this project done now so I can finish in 3 years. Like, no, you'll be here for 5 years. That's not going to change. Whereas with the postdoc, you're in it as long as it takes you to be productive. So I definitely feel far more pressure here than I did in my PhD.
Yeah. I think that's the right idea, though, to not necessarily kill the project, but put it on the back burner and let it linger in your mind. And, you know, when you have time and when the appropriate tools become available, then you can act on that. But yeah, sometimes it's just not the right time, I think.
Yeah. And it's been the same thing too, that I, you know, in getting more into the pox virus field, I've also like come up with other grant ideas where I'm like, this would be really interesting, but it's like I just need to like cut. You have to put on the blinders a little bit. You know, it's really easy when you get into something and you're excited about it to be like, what if I do this and I spin out this and I spin out this, and it's like at the end of the day, I'm one person. I can't do 7 diverse projects all at once. So it's like, okay, I just need to catalog this. This is an interesting idea. Like, let me circle back to it later.
I know a lot of undergrads, grad students, and postdocs listen to the podcast. Do you have any advice for these people who may be considering pursuing a career in academia and in immunology and virology?
I think people are aware of this, but it doesn't necessarily get a lot of attention. You know, I think that we all know science is hard, and we to some degree kind of tell ourselves like, okay, I just need to like put my head down and like push through. And you tell yourself that in your grad years, you tell yourself that in your postdoc years, and the burnout is tough. So like, I think there's a lot of factors to the labs you're in that people may say that, oh yeah, I can just like push past that. You know, like you may have a mentor that you don't fit with very well. And you may say to yourself, well, I'll just power through. And that can work. You know, I think it's a workable strategy, but also like, it's not a pleasant strategy. So I definitely think take all the factors of the lab as a whole and really consider each part. You're never going to have a perfect mentor, but find mentors that can meet you where you are and that are also willing to be flexible. You know, when I was between picking, you know, Shane or John's lab, you know, at the time, I felt like I needed a lot more hands-on. And, you know, I think John is a really great guy. I think he does really great science, but John is just much more of a hands-on mentor. He has a harder time of pulling back. And so there were times that, you know, we would butt heads over direction of a project and how to handle analyses. And when I was looking at Shane's lab, when I rotate through the lab, I would get Shane's time, but Shane was just much busier. And so, he's in higher demand. But there was an environment that supported people in the lab. There was a lot of great, fantastic postdocs in that group. And there still are, that I could go to with pretty much any question. They could help support me. At the time, though, I kind of said, well, I think I want somebody much more hands-on right now. And so I went with John based on that. And by the end of it, it was like, okay, now I need someone more like Shane for the end here. And so you're never going to have a perfect mentor, but in John's lab, I also had, some really great people that, postdoc, senior scientists I worked with, as well as the junior faculty member. Both of them were really fantastic, and they helped me in different ways in giving the mentorship that I needed. And so, particularly on the mentor side, acknowledge that you're never going to find the perfect mentor, and try and find one that's able to kind of meet you where you are, and especially recognize that you're going to grow. So like you're going to need different things over time. And so as that occurs, put together kind of a panel of other mentors. I brought in Raman Haradi as a co-mentor because Mark is far more clinical science. He doesn't have that hardcore basic immunology that Raman really did a postdoctoral experience, John Weir's lab doing. And so there was a lot more of that expertise with Raman, and I needed that for the project and where it's going. And so that's when I brought him in, and it's been really helpful in being able to have that other scientific mentorship, but also it's just a different mentorship style too. So it's just good to build that community and build that network and have that support network on the mentor side, but also on the community side, your colleagues, your friends, your cohort.
Curious about the bringing on the second co-mentor to follow up with that. So during your postdoc, you were originally just full postdoc in the Mulligan lab, and then later decided you wanted to bring someone else on. And it was not just an on-paper thing. It's like you're going to their lab meeting, you're meeting one-on-one, it's full co-mentorship.
Yeah. So I brought him on actually earlier this year after I was in the lab for about 2 years. And it was because I was hitting the point where we needed to look more at the cellular side of this question. And there just wasn't the expertise within the lab and what Mark had previously done to be able to support the in-depth studies that I needed to be doing for this project. And so I went to Raman and I said, look, this is the position I'm in. I really need this extra support. And I brought him on, and yeah, I'm basically on paper, I'm in both labs now. And I go to both lab meetings. As part of Raman's group, I'm integrated into a few different other networks and stuff. And that's been also great, getting even deeper feedback from other groups that are in complementary areas of immunology. And so it's been really valuable having this expanded network, especially because, the Mulligan lab is on the smaller side right now. Within the lab itself, within the lab proper, I'm the most senior person doing research. So I think as a postdoc, you can pick things up a lot quicker, but it's still really nice having somebody who has hands-on experience in a technique that you can go to and say, can you help me troubleshoot this? So, and that's really what I've gotten from Raman's group.
That's great. I might be considering something similar.
Yeah.
So my last question here. So this is the Inflammatory Content podcast. I have a question that I like to ask people now, and it is, do you have any hot takes about science or virology? Anything that you believe other people think is a bunch of bogus?
Yeah, I mean, I mentioned earlier the whole question of the clade 1 Mpox. I think that we are just absolutely wasting time. I think that we are throwing valuable time out the window right now. And it's been a little bit aggravating. We've had a number of grant reviews come back, and also, some other smaller things that have come back to us where we're proposing Mpox work. And up until a few months ago, you basically had a luck of the draw, whereas either people that are like, yeah, okay, this seems important, versus other people are just like, who will ever care about Mpox? And as this clade 1 outbreak has been going on in the DRC, you've been getting a little bit more people that are just like, okay, maybe it is important. But there's still people that are just like, who could ever possibly care about this? And that is wildly frustrating. And you never want to wish for an outbreak. Like on some level, if you're an infectious disease, you kind of don't want your work to be hyper relevant because it means that some pathogen is ripping through the population. Like, ideally, if it's something that's everywhere, then okay, it's something that is maybe a low grade non-pathogenic virus or something. But I don't want clade 1 Mpox to spread through the population, but I just look at the response and I see the people that are saying like, who cares about Mpox? And it's like, well, give it a year, give it 2 years. We're going to have the exact same thing happen that we had with clade 2, and we're going to have clade 1, and it's going to be far worse. And we could have stopped it. So I find it aggravating when people are just underwhelmed by the whole field of poxviruses. But who knows? Maybe that'll change. Hopefully for good reasons, not for bad ones.
That's a good hot take. I like it. All right, Aaron, any final things you want to say before we wrap up? It's really great catching up with you.
Yeah, no, I think that covers it. Yeah, it's been great chatting. I think it's really always fun getting to have these longer discussions about science and both the research, but also kind of science, in the state of the work from a career side. Because I think too often we like to put our heads down and just tell ourselves like, yeah, you just have to struggle, and then eventually you'll get to the other side of it. And it's like, well, do we have to? Can we do it better?
All right, Aaron. Well, hopefully we'll see you in New York sometime in person.
Yeah. Always happy to have people come through. We'll share a beer, talk some science.